Hello, my name is Sabaoon and I'm a research assistant at the Precision Medicine Lab, Peshawar, Pakistan. Today I'm going to talk about CMOS, which is an encyclopedia of multi-omic stacks and their respective cell lines in head and neck squamous cell carcinoma. In cancer research, cancer models are utilized to better understand the tumor microenvironment. And in this process, cancer cell lines are a major tool to understand cancer on both genetic and epigenetic levels, and also the cellular pathways that are involved in cancer. And similarly, it goes for HNSCC cell lines. These cell lines are either established through cell culture assays such as 2D monolayer and 3D spheroid cell culture systems or the explant cultures. And till date, globally, hundreds of HNSCC cell lines have been established. Although the data on these established cell lines are present throughout the databases such as Cellosaurus, CCLE, Cell Model Passport, Gene Expression Omnibus, but this data is scattered and it needs to be cataloged. We got our inspiration from a study conducted in 2006 by the lab of Dr. Stephen Lai, where they collected data on 428 HNSCC cell lines. And since then, there has been a revolutionary advancement in the technology, and now the literature lacks a catalog with all the information related to HNSCC cell lines. So that was our major goal. Following was our approach for conducting the study, where we collected data on demographics, primary cell culture protocols, mice xenograft models, reported mutations, and the multi-omic datasets that are present out there. So till May 2020, we collected data for 979 HNSCC cell lines. Starting with the demographics, we collected data on the tissue type, country and ethnicity, non-substance abuse history, treatments performed, and other parameters such as age, gender, TNM stage, etc. Similarly, for the reagents used in primary cell culture protocols, data was collected on primary medium, supplements, antibiotic or antimycotic solutions, mice xenograft models, and other parameters such as the storage condition, carbon dioxide rate, doubling time, etc. For the reported mutations, various databases were searched, such as Tumor Portal, CBio Portal, Cosmic, and various relevant databases. And we came to know by the end that TP53, CDKN2A, PIK3CA, and Notch1 were the most occurring mutations in the HNSCC cell lines. So the most exciting part of our study was collecting a lot of multi-omic data sets for these head and neck squamous cell carcinoma cell lines. And we collected data for 10 omes that included expression profiling by array, non-coding RNA profiling, methylation profiling, RNA-seq, ChIP-seq, proteome, whole exome sequencing, whole genome sequencing, metabolome, and SNP array. And over here the blocks represents cell lines that were being used in similar studies or in large scale screens. And the most exciting part was that for all the 10 omes, data was available in 3 cell lines that were Cal27, FADU, and HSC2. And once we collected all that information on HNSCC cell lines, we shaped it into cells with multi-omic stacks, which is a database that we called CMOS, and it can be accessed through the given link. So we established a comprehensive catalog of 979 HNSCC cell lines. We collected data on the demographics, reagents used in primary cell culture protocols, xenograft models, reported mutations, and all the available multi-omic data sets out there. We cataloged it, and we gave it a shape of a database that we call CMOS, and we hope that this database, it will be a benchmark for the young researchers and the scientific community that are who are working on head and neck cancers for breakthroughs in HNSCC research by using computational and experimental analysis. These are my acknowledgements. Thank you.